Hello everyone, thank you for joining today's webinar, Scalable Lentiviral Vector Manufacturing Using Suspension Cell Line. My name is Jie from GenScript Europe, and I'd like to welcome everyone for today's event. Today's webinar will be approximately half an hour, and it's a pre-recorded webinar to avoid technical difficulties. In case you have any questions during the presentation, please submit your questions in the comment section, then we will collect your questions and get back to you via email individually. I'd like to introduce our speaker, Dr. Xuan, who has more than 7 years of experiences in the process development of lentiviral vector and CAR-NK. Please welcome Dr. Xuan.
Hello everyone, welcome and thank you for dialing in for our webinar today. My name is Chunling Xuan. I am an R&D manager at GenScript ProBio and responsible for viral vector manufacturing process development. Today I'm going to talk about scalable lentiviral vector manufacturing using suspension cell line. Okay, let's begin. Today's webinar includes the following 4 parts. Firstly, I'll introduce the market perspective of lentiviral vector in cell therapy, and I'll give you a brief introduction about the basic structure of LVV and how it works. Then, followed by manufacturing optimization strategies, I'll also share some successful data. Finally, what we can help to accelerate your cell therapy project will be included. Viral vectors provide an efficient means for modification of eukaryotic cells, and their use is now commonplace in academic laboratories and industry for both research and clinical applications. This table shows the drug delivered using viral vectors, showing the repeat growth of viral vector drugs in the past 6 years. As you can see, 4 products using lentiviral vector as delivery vehicle. From the perspective of market demand, you will see the increase in demand for LVV produced by suspension systems. Cell therapy accounts for a large amount of market share and is increasing from 2018 to 2020. Over 40% of cell therapy programs in clinical phases, larger amount of LVV required for clinical phases. For cell therapy application, increasing number of cell therapy programs are targeting solid tumor, which will require larger amount of LVV. The second part is basic introduction of lentiviral vector. As you all know, lentivirus is a subclass of retrovirus that has been successfully used in the clinic and are increasingly being used for non-clinical applications. They are capable of stably transducing a broad range of mammalian cell types, including non-dividing cells with high efficiency. Utilizing this feature of lentiviral vector allows for the permanent expression of a gene construct, such as a siRNA or protein coding sequence, in a population of cells or on a single cell level. Lentivirus contains 2 copies of linear, single-stranded RNA of 7 to 12 kilobase pairs in length encoding the gag, pore, and envelope genes. Advantages of lentivirus include a large genetic capacity and the ability to transduce both dividing and non-dividing cells. The first lentiviral vector system consisted of 3 plasmids: the packaging, envelope, and transfer plasmid. The packaging plasmid carried a mutated HIV provirus that couldn't package itself due to a few missing proteins. The HIV genes that do remain are very important for viral production, like GAG, TAT, and REF. A different viral envelope protein, usually from VSVG due to wide effectivity, is often used as a substitute for HIV envelope. To further enhance effectivity, the third generation system was created, which differs from the second generation in two key ways. First, the packaging system is split into two plasmids, one encoding REF and one encoding GAG and POM, decreasing the number of recombination events necessary to create a competent virus. Second, TAT is eliminated through the addition of a chimeric 5' LTR containing a TAT-independent promoter. Here you can see the procedure of lentiviral vector production and transduction of target cells. Firstly, HEC293T producer cells are co-transfected with the lentiviral vector plasmid and 3 packaging plasmids, and then lentiviral vector particles will be assembled and secreted in the supernatant. The vector particles can be used for transduction of target cells, which results in viral DNA integration into the host cell genome. After talking about the basic information about the lentivirus, in the next part, I will talk about the optimization in the suspension lentivirus vector manufacturing. As I mentioned before, lentiviral vectors are increasingly utilized in cell and gene therapy applications. However, the manufacturing of cGMP-grade LVV has been a challenge for early-phase clinical trials and is even more difficult for commercialization. Today, standard LVV are made using a third-generation production system that relies on the transient transfection of HEC293T cells. This transient transfection method can generate high-titer LVV. However, because of the adherent and serum-dependent nature of the HEC293T cell line, cGMP LVV production is limited due to the need to use multi-stack plastic tissue culture vessels, which increases the amount of handling, personnel, and consumable costs, and production time. Furthermore, the use of animal-derived serum to maintain the HEC293T cell line increases the risk for contamination by adventitious viruses. In addition, obtaining large enough lots of serum can often be challenging and costly to acquire. To address these challenges of industrial-scale cGMP lentivirus manufacturing, many strategies use HEC293T derived cells that are adapted to grow in suspension with serum-free medium. This suspension system produces high titer in supernatant and a high batch yield, but the HCP is relatively high, which will pose challenges to the downstream process. Next, I'll introduce the challenges encountered in the lentiviral vector process development. In viral vector manufacturing, care must be taken to ensure maintenance of viral vector potency. Since the lentivirus is an enveloped virus, it's sensitive to the detergent, temperature, shear force, and changes in pH. Such drawbacks should represent major challenges in the development of robust and fast downstream processing strategies. Large-scale production using cGMP methods comes with a set of challenges that must be considered as more clinical trials using lentiviral vectors receive regulatory approval. One important consideration in designing a cGMP-compatible process is the need to have a manufacturing protocol that produces consistent lentivirus for multiple cGMP productions. During transient transfection, DNA must be mixed with a transfection reagent and then added to the bioreactor. Fluid dynamics in different bioreactor volumes are a concern, as there is a natural window of time in which mixing and subsequent addition to the cell should be achieved to maintain consistent transfection efficiency and the production yield. For downstream processing, similar processing time can be achieved by scaling up volumetric flow rates for chromatography or filtration steps. GenScript ProBio one-stop solution from plasmid to lentiviral vector can solve these industrial bottlenecks. And now, I want to share some actual cases about what we have done to optimize our lentivirus manufacturing process. There is an increasing demand for biopharmaceuticals produced from animal cell culture process. To optimize large-scale clinical-grade lentiviral vector production, we developed an improved production scheme by adapting HEC293T cells to grow in suspension using commercially available and serum-free medium. We have successfully selected a monoclonal HEC293T cell, which is successfully adapted for robust growth in suspension culture using a serum-free medium. This cell line is named PowerS293T. Our PowerS293T has stable produced lentivirus in 22 passages. We also optimized the transfection parameters during the upstream process. Since the transfection reagent is positively charged, the plasmid could break through the electrostatic barrier on the cell membrane and enter the cell. Thus, during this process, plasmid ratio and concentration, transfection reagent, and cell density are the key technological parameters of lentivirus transfection. To test the stability, we used transfer plasmids containing different GOI to package the lentivirus. 5 batches of data showed that the cell line is very stable. Then, we explored our method in scaling up the manufacturing process. We scaled up from 30 ml shake flask to 10 liter bioreactor. The crew titer showed good consistency from small scale to large scale, both in shake flask and glass bioreactor. We compared the cell proliferation and availability in the shake flask and the 2 liter bioreactor and the 50 liter bioreactor. The results showed that the cell growth in the 50 liter bioreactor was stable. The crew titer showed good consistency from shake flask to 50 liter bioreactor. In this slide, we show the CPP and CQA that should be paid attention to when optimizing the downstream process. Since the diameter of lentivirus is 80 to 120 nanometer, there will be a lot of loss during sterile filtration. Therefore, it is necessary to control the turbidity and the particle size of lentivirus to improve the recovery rate. Anyway, these CQA should then be maintained throughout the product life cycle by carefully controlling and monitoring those CPP that may affect them. 5 batches data show that The downstream process is stable. Okay, after talking about how to optimize upstream and downstream process, I'd like to introduce the highlights of our proprietary suspension cell line, PowerS293T. PowerS293T is ProBio proprietary lenti suspension cell line. By comparing with the top vendor's commercial cell line, the titer of our cell line is slightly higher. Using different GOI, the core titer is about 10 to the power of 7 TU per ml. GOI 1 to 6 are different transfer vectors of CAR-T or TCR-T. The second highlight is its good stability and scalability. Stable titer from 2 liter to 10 liter in 2 projects. More than 3 batches of stable titer in Project 3 and Project 4. I think everyone must be very concerned about the efficiency of transduction of T cells by lentivirus produced by suspension cell line. Compared with the lentivirus produced by the adherent system and by other commercial suspension systems, the lentivirus produced by our PowerS293T is more efficient to introduce genes into mature T cells. The first highlight is high output and low cost. Compared with the adherent system, the core titer is increased by 10 times, the yield is increased by 5 times, and the cost is reduced by 50%. Last but not least, I'll briefly introduce what we can help now. GenScript is a global biotech company and is divided into 4 segments, and we are the GenScript ProBio, focused on biologics CDMO. GenScript ProBio proactively provides end-to-end service from drug discovery to commercialization with professional solutions and efficient processes to accelerate drug development for customers. Throughout these years' development, we have completed 60 plasmids and various CMC and clinical projects, 8 IND approved from NMPA and FDA, and 3 mRNA projects IND approved, and we have conducted over 20 CMC projects. We also provide plasmid and viral vector service for over 30 big pharma and leading biotech. Currently, we provide from plasmid manufacturing to LVV CMC study and manufacturing services, and we can support your projects in the whole drug development circle. We have ProGrade for preclinical study, CMC study for IND filling, clinical GMP grade for early phases, and cGMP grade for late clinical phase and commercialization. In order to accelerate your cell therapy projects, the lentihyperplasmids, which have clear history and without IP issues, are immediately available and ready to support FDA IND or early phases clinical trials. The lentihyperplasmid DMF has been registered with severe FDA. The advantage of the lentihyperplasmids is to save the time and cost in development of 3 hyperplasmids and simplify regulatory documents. In summary, our LVV service has the following 3 advantages. First, our cell line PowerS293T has independent property right, which is cost effectiveness for license, royalty-free and maintenance-free. Second, the production of lentivirus is high in yield and low in cost. and the lentivirus has good efficiency in transducing T cells. The average yield is 10 to the power of 7 to 10 to the power of 8 TU per ml. Third, ready-to-use third-generation lentiviral system with FDA and the approved case. GenScript ProBio has an excellent track record in lentiviral vector projects. 3 IND approved from NMPA and FDA, and 10 IND projects are ongoing. We have completed more than 10 GMP batches for clinical trials. Thank you for your attention. That's all what I've got for you here today. Thank you.
Thank you, Dr. Xuan, and thank you everyone for your attention. Please note that this is a pre-recorded webinar, therefore your questions will be shared with our speaker, Dr. Xuan, after the session, and you will be answered via email. If you haven't submitted your questions during the presentation, please send your questions to eumkt@genscript.com anytime. I hope to see you again in our next webinar. Thank you and bye.